Imagine a protein so chaotic and destructive that it fuels 3 out of every 4 cancer cases worldwide. For decades, scientists have known about this molecular villain called MIC, but they've been powerless to stop it. MIC normally helps cells function properly, controlling how genetic information flows from DNA to proteins. But in cancer cells, everything changes. MIC becomes hyperactive and unregulated, acting like a steroid that supercharges cancer's rapid growth. MIC is less like food for cancer cells and more like a steroid that promotes cancer's rapid growth, explains Professor Min Shui from the University of California, Riverside. The challenge has always been MIC's structure, or rather, its complete lack of one. Professor Zhe describes it as basically a glob of randomness. Conventional drugs rely on targeting well-defined protein structures, but MIC doesn't have one, making it nearly impossible to control. Until now. Researchers at UC Riverside have developed a groundbreaking solution. They created a specialized peptide compound that can bind to and suppress MIC's destructive activity. By carefully manipulating the peptide's shape and rigidity, they achieved what's called sub-micro-molar affinity, meaning the peptide binds to MIC with the strength and precision of an antibody. The results are remarkable. The team improved their peptide's binding performance by 2 orders of magnitude compared to previous attempts, bringing them significantly closer to developing an actual drug. Currently, they're working on delivery methods using lipid nanoparticles to get the peptide into cancer cells effectively. This research, funded by the US Department of Defense and the National Institutes of Health, represents a potential turning point in cancer treatment. After decades of being untouchable, MIC may finally be within our grasp. The study appears in the Journal of the American Chemical Society, and more on this research can be found from a link in the description to the full article on Earth.com.